To be our assignment that we'll refer to as the segment polarity gene assignment. And it is about setting up the anterior and posterior compartments in each segment of the insect. Okay. The segment polarity genes are also called segmentation genes. That's what our textbook calls them. Segment polarity genes is the better name for what we're going to be working on. Okay, so it's setting up those and the anterior posterior axis, the animal. Okay, and it's a drawing assignment, and so I put some materials outside of my office, and there are tables nearby, so you don't need to go out and buy anything. If you have some colored pencils and markers at home or other kind of stuff, you want to try to make something that's visually memorable, to help your learning now and also your retrieval when you're remembering things for the exam. And so this assignment is about the segmentation genes, and I've described how it'd be nice if the author made this as two figures, because we have really two separate final stories. This is our first story, and it's setting up the expression of the Hox genes. And we've learned that there's a cascade of gene expression starting in the germ cells in the female ovary, and we get a series of gene expression in the embryo of maternal factors and then zygotic factors, going from the genes like maternal genes like bicoid to zygotic factors like the gap genes, the parallel genes. And those lead on to the bottom of the pyramid where we have Hox gene expression, and we set up the identity of the 14 segments of the insect in the anterior posterior axis. And we've talked a lot about Hox genes now. This is really a separate bottom of the pyramid. This is the story we're telling today. So this has, at the bottom of the pyramid, we have the segment polarity genes or segmentation genes. And the story above it is comparable. There's a set of maternal factors that are turning on these zygotic factors to set up the segmentation genes. And it's just too bad that our author doesn't show that as really two separate stories. And in the case of the first story, Hox gene expression, we told a story along that timeline because the emphasis is about, well, how does this get all organized and how did the dominoes fall leading to Hox gene expression. In this, so that's story number one, and then we're telling story number two now. And in story number two, we don't bother telling this story because it was a little bit of a kind of repeat, and it's an introductory course, so let's move on to the new stuff here. And the new stuff is to draw a picture of what's happening in the segment polarity genes. And so that's our goal, to draw that out. And I'm going to start drawing it like this. So I set this up, and up in this corner, up in this corner, it's good to draw a little picture of what your goal is. And so here's some segments, like in a caterpillar. So there's a little happy caterpillar cruising along, and that first story is about how each of these segments have different identity based on Hox gene expression. Okay, what we're going to be looking at here is each of these segments has a organization within the segment where there is a boundary here, like that, and that's called the segment bound. No, it's called the compartment boundary, like that. So we're going to have a compartment boundary in each segment, and it's an invisible line, and all the cells on the anterior side of it are in the anterior compartment, and all the cells in the posterior compartment are posterior cells. And so down here, well, right here, we're going to have a cell here, and down here, our goal is to write a cell down here like this, and that's going to be an anterior cell. We're going to draw what's going on in the anterior cell, and then we're going to have this be the compartment boundary. In our book, the book describes this as the parasegment boundary. And that's because when the boundaries first get set up in the embryo, it's actually these parasegment boundaries that get set up, and only later does the animal create the more familiar observable segment boundaries. Okay, so that's why the term parasegment was invented. So we're going to have an anterior cell here, and then right next to it we'll have a posterior cell. And all the cells in the anterior compartment are anterior cells, and all the cells in our posterior compartment are posterior cells. Okay, so that's the drawing we're heading to. We're going to give a little background before we get there. Oh, before we leave this, we're going to especially emphasize what's happening in the posterior compartment cell. And the idea is, what genes are expressed to have this be a posterior compartment cell? In other words, what genes are expressed to give it that identity? The anterior cells are anterior cells because they're expressing a different set of genes. And it'll be a comparable set of genes, and so we won't learn all of the story in the anterior compartment just because it's a parallel story, a comparable story to the posterior. So we'll emphasize this, learn this, and we'll talk about the communication across the compartment boundary in order to understand what's happening in the anterior compartment cell also. Okay, so that's where we're heading, and it's this segmentation gene story from the book, or the segment polarity gene story. And from our handout, this story really emphasizes these genes here. And there are going to be more genes, but we just start out especially emphasize those, and we build out from there. So those are the genes that we want to know the most about, but then we want to learn more, and we're going to have a list. So there's more to learn than that. It's just the starting point and the most important part of it. Okay? All right. And so here's some information about segment polarity genes. And here's, to start with, here's the phrase that our book likes to use. A segmentation gene is a generic term for a gene whose function is to specify a tissue pattern in each repeated unit of a segmented organism. Okay? We're going to use this term for that, segment polarity genes. Segment polarity genes help to define the anterior and posterior polarities within each embryonic parasegment by regulating the transmission of signals via the Wnt signaling pathway and hedgehog signaling pathway. Great, okay? So that's where we're heading at. And, but we already know a lot about this. And so I just want to review this idea of how gene expression determines the identity of cells. And we have some examples. So our first example, we're going to talk about our muscle genes from our muscle differentiation videos at the start of our course. And those images there showed that the myogenin gene was activated, and the myogenin gene encoded a protein that's a transcription factor that would bind to the target genes that are muscle-specific genes. So from that, we get this concept that muscles are muscles because they express muscle genes. And we're using that as an example. That's a general example. So brain cells are brain cells because they're expressing brain genes, and livers are livers because they're expressing liver genes. Remember, every cell has all the genes, but they're expressing different subsets of the genes, like that. And so scientists refer to these as muscle-specific genes. They're found in muscles, and they use that as a rule to determine if a cell's a muscle. They check to see if it's expressing muscle-specific genes. If it is, that must be a muscle. That's our definition as molecular geneticists. That's our definition of a muscle, is it's making those genes. And same for these other tissues. In general, scientists refer to these as tissue-specific genes. And these are genes that are expressed when a cell reaches its fate, right? It's fated to be a muscle cell, and when it reaches its fate, it's expressing those tissue-specific genes that its fate dictated, like that. And these tissue-specific genes are expressed during differentiation. That's when a cell gets to the final point in development and becomes its fate, becomes the muscle cells, and it does that by expressing those specific genes. So we have this idea from those is that there are these tissue-specific genes that are associated with a cell's fate and all the different tissues, like eye lens cells and salivary gland cells and so forth, kidney cells. They all have different tissue-specific genes, and so that's an important example. Then the next example we had was, and we've had other examples too, but should be a memorable example. We said brachyury is a mesoderm specifier. So we had this image of a belt and a belt buckle, and brachyury is a transcription factor expressed in those cells, and the brachyury would activate expression of genes that are mesoderm specifying. Now, the mesoderm is not differentiated. Those cells have a long way to go in development. They're fated to form bones and muscles, and that's not yet, right? So now we have a thing that a cell is always expressing some cell identity type specific genes. Like this is supposed to be mesoderm, so it's going to be expressing mesoderm genes. This is the ectoderm up here. That's going to be expressing some ectodermal genes. This is the endoderm down here, like that. So now we have that addition thing, not just differentiated genes, but even genes along the developmental pathways are expressing this. Then we had a third good example of this story, and that is these stem cell identity genes. And we talked about these scientists, and especially Yamanaka, who discovered that if he took a differentiated cell and he forced it to express stem cell identity genes, that cell would become a stem cell. Because, like the mesodermal cells or the muscle cells and these other cell types, a stem cell is a stem cell because it's expressing certain genes. And we call those stem cell identity genes. And Yamanaka found that it was just 4 genes that he needed to force the expressor of to misexpress them to turn any cell into a stem cell. And so we have this term, these induced pluripotent stem cells. And these are cells that could have been differentiated cells and from your body, like just a skin cell, and we misexpress 4 stem cell identity genes, and those cells will become stem cells, and we call those special induced pluripotent stem cells because they are artificially constructed. They're not embryonic stem cells, they're not adult stem cells, those are natural stem cells. These are artificially produced, like that, okay? And I just pulled this slide from our earlier lectures, and so I had it on there, but it is, the concepts used to build our clay model video are all brought together here like this, okay? So we have those examples of how gene expression determines the identity of cells. Major concept, okay? And so that's going to be happening here, and we're going to show that in our figure. So the posterior cell is going to be expressing some what you could call posterior cell identity genes. It's not a differentiated cell, it's very early in the embryo, and we're going to have a list of them that we'll be working on, like that. Okay? And so that's what your drawing is going to look like. Okay? So next I'll just show you some figures that are the basis for our drawings. Okay? So here's the end. Here the book has really 2 images that show this story in kind of summary. That's a first image, and here's a second image, and we aren't going to talk through those in detail, but you should look at them in that portion of the book, and if you have questions about what these represent, I'll just add something here, just to show you what this figure is depicting. And so what this figure is depicting is, it's actually just showing in the syncytial blastoderm, so before cellularization takes place, about 2 hours after fertilization. And The embryo is about 700 nuclei. It's a very specific number. It's about that many. And they're all around the outside of the embryo. And so this is like take an embryo and slice it in half the long way, and you would see this ring of nuclei. And what this figure is it's just taking a strip of, it looks like about 16 of those nuclei. So it's taking a picture of 16 nuclei, and that's what's shown there. And then it's showing those same nuclei 3 different times. And each time it's showing the expression of a different set of genes, like that, okay? And so we have segment boundaries here. So here's a segment. And so it's showing 4 nuclei just because that's how many are involved in each segment at this early stage. And of the 4, actually 3 of them are anterior, and only 1 is posterior. And this is our drawing right here. Our drawing is just this, and we're going to show these genes in one drawing and more. So just a little background on that figure. And at the time of our drawing, they're cells. So our drawing is just about an hour later after cellularization has taken place. Okay? This is from a different book, and I like this figure because it shows the cells bigger, and this is really the drawing we're going to do. Here's an anterior cell, here's a posterior cell, and this is literally the drawing we're going to do and add to it. Okay, so here we go. So let's do this. So the first thing is that the anterior cells release Wnt. And so they have to be making it. And remember, we had a drawing assignment where we said, oh, the Wnt signaling, this must be a source cell, and this must be a target cell. Back to our Wnt drawing. This is our Wnt drawing to start with, and it's literally our entire Wnt drawing. We'd like to show that the Wnt gene needs to get activated in here, and we need to get Wnt RNA and Wnt protein made, and then have it secreted. I might not use exactly consistent colors here, because I'm doing it quickly, but try to use a variety of colors and in a consistent color pattern so that you can see and learn better, okay? So then for this target cell to be a target of the canonical or the typical Wnt signaling, it's got to have frizzle. And that's our first thing. To be a posterior cell and have that posterior identity, one thing you need to be doing is you need to express the gene frizzled. And so we know from our Wnt drawing how that gets done, right? So through signal transduction, frizzled uses disheveled, activates disheveled to block GSK from targeting beta-catenin from destruction. If there's any beta-catenin over in this cell, it's getting destroyed. This cell, the beta-catenin is stable because of the Wnt signaling. And beta-catenin gets to do its job, and its job is to bind to target genes that are initially repressed by this repressing protein TCF, and you can just review your Wnt drawing. It's blocking that. We had that Wnt drawing on our first exam, and everybody did a great job. And beta-catenin binds here and overcomes that repression and activates these target genes. So these are beta-catenin target genes, and they're getting activated. Those genes are present in the anterior cell, right? And they're being repressed because TCF is there repressing them, and beta-catenin is unstable. So this is a step to be a posterior cell, is we got to activate this, and it's that Wnt signaling signal that says be a posterior cell. All right, So what are some of these target genes? Well, one target gene is hedgehog. And hedgehog is the fruit fly ortholog of sonic hedgehog, and it's going to get released like that, and it's going to bind to its receptor here in the anterior, which is called patched. That's the receptor, and there's going to be a response, and the response is B anterior. And to be anterior, have to activate a set of genes. And we're not going to emphasize that, but the set of genes have to include make Wnt. That's part of being a anterior cell. Also, make sure you keep making patched like that. Okay? So on our list of genes we need to make to be posterior, posterior identity also involves hedgehog. And that's an important concept in our course. In having this identity during embryogenesis, lots of times the cell is actually producing signals to help another cell with its identity. So part of having the identity of being I'm a posterior cell is to express the hedgehog signal that tells cells they're a anterior cell. Patched, right? Patch is a protein, and this cell doesn't have it. So part of being a posterior cell is repressing probably some gene expression, not expressing it or repressing it. And so that brings up our third gene example, and that is another target gene, is a target gene called engrailed. And engrailed codes a protein that's a transcription factor. And so let's put that here. And engrailed helps activate posterior genes. So part of having a posterior identity is to express posterior genes, and some of those genes are transcription factors, not just engrailed and beta-catenin, but others as well that help express a variety of posterior genes. So activate posterior, and also those transcription factors repress the anterior identity genes, right? Because they have it. This cell has patch, this cell has Wnt, but it's not making it because it's repressing it. So that's our next example, is just that. And that's true in all those other examples, that in those induced pluripotent stem cells, for example, of the 4 genes that need to be misexpressed, the Yamanaka factors, all 4 of them are transcription factors, because transcription factors can turn on a whole set of target genes. And so we want that on our list to especially appreciate that this cell is expressing a set of genes, including specific transcription factors. Can we think of another example of that? Yeah, sure. Remember, in muscle differentiation, in order to express the muscle-specific, tissue-specific genes, we turn on myogenin. Its job is just to be a transcription factor. What kind of transcription factor? In that case, it's one that is going to help activate muscle-specific genes, like that. And engrailed can help activate a set of posterior genes, and engrailed also functions at other times in development, in other contexts, to activate a different set of genes. So there's some flexibility in some of these transcription factors because they have a lot of targets. Okay, great. All right, and so all this so far should fit in really well with the kind of learning that we've been doing in our course. And then the next gene, though, is a new gene, and for us, and they're called cell adhesion molecules, and they are proteins on the surface of a cell. And we'll learn a lot about them in Chapter 7. And Chapter 7 is essentially, I could call it the cell biology of development. It's a very cool chapter. So here's the cell adhesion molecule, and I'll just draw them like this. So they just are on the surface of the cell, and they have a shape to them. And these like to hold hands. So there's another posterior cell, and they're holding hands. Just like literally like a team of soccer players staying together because they're all holding hands with each other. And we don't need to learn the names of them now, but we'll learn some of the really famous ones in chapter 7. Because we'll see that even the most distantly related animals, and those would be sponges, and maybe there aren't even animals, but let's say they are. Sponges have these orthologs of these famous cell adhesion molecules, and that's how they set up their very simple diploblast body. They just have an inner layer of cells and an outer layer of cells in sponges. And they do that by holding hands, by having the sort of inner compartment and outer compartment of cells. Just to draw it, just to give a kind of an example of this, is that to be an anterior cell means to express some different anterior cell adhesion molecules. So these guys are holding hands like that. Like that. Our book shows that if that happens, the two teams stay segregated. In the wild type animal, they're physically segregated because they're holding hands with each other. The anterior with them, each other, and the posterior with each other. And that's how we get these two populations of cells that are right next to each other, right? They are right next to each other to become completely different. And our book shows, let me just see if I can find this slide. Our book shows this idea that we have a natural compartment boundary. And so these would be anterior cells and posterior cells. And if we have a mutation in engrailed, we failed to establish that really distinct difference in gene expression, right? Because engrailed normally helps activate posterior cells and repress anterior cells. And if that doesn't happen, we just don't get that good, strong team separation. Why not? Because we have problems with the expression of the cell adhesion molecules, and they don't hold hands very well like that, separate. Since we're on this, just to show you this is just showing that in wild type wings, the anterior cells stay in the anterior region. It's just showing a genetic test of that idea. So the cell adhesion molecules are, I think I sort of messed up some buttons here, but we're fine, okay? So back to here. So number 4 are these cell adhesion molecules, which will be a big part of what we want to talk about. And then we're going to add another one, and that is going to be that this boundary we just saw from the book, the boundary starts to form when the cells start to form about 3 hours after fertilization. And at that time, each segment is just 4 cells wide. So that's one segment. And so there's going to be a lot of growth and a lot of cell division. And what this compartment boundary does to serve is it serves as like a reference, like a landmark, to allow cells to become different over time during development. And the way I describe it is that you've seen when somebody's getting ready to construct a new building, there'll be these surveyors out, and they'll have like a sort of looks like a binoculars on a tripod, and they'll be making measurements. And what they're doing is they're measuring and determining a line that would be a level line, even if the construction is going to be happening on a hill. They determine a level line to make sure that when they make the foundation of the building, it'll be level, and the building will go straight up into the air as they build it, even if the building is being placed on a hillside. So this boundary is similar to that. During development, like during the construction of the building or the construction over time of the segment, in each segment of the animal, This is providing a reference and of the position. And we're just going to use one gene from that, and that gene is called DPP. It stands for decapentaplegic, and it's actually one that's listed in our handout as part of the third story. So we'll meet this and get to know it well at that time. Here we'll just get a brief introduction to it. And so DPP is expressed in these cells, and DPP is a secreted signaling factor. DPP is a homolog of BMPs. And we haven't used that term homolog very much, but we talked about it. Homolog is just a more kind of like casual use of the term ortholog. And it's being used here because these are so distantly related. They're clearly closely related, but we're not exactly sure if they're like orthologs or not. So we say homolog. One of the reasons is because when we say BMP, we're talking about the names of genes in vertebrates. And in our bodies, I think we have like 30 or 40 different BMP genes. They're numbered like that. And so we have like BMP4. And so it's a little bit hard to determine what you really want to call that an ortholog. It's like doing the same type of thing, okay? But it's a secreted signaling factor. Together, these are, our book likes to say, they're in this TGF beta superfamily. And we've had lots of examples of that, from BMP to also XNRs are in the TGF beta family members, and VEG1 is in there. So, we're meeting several of these guys, but just DPP. And DPP is said to be a long-range signaling factor. The Wnt and hedgehog are short range. They signal only just over a few cells diameter. And DPP is more stable and spreads out further. So as a result, let's just do this, and we will add a little bit to this. So let's say that if we looked in this segment and we did that thing where we drew a triangle, we would say there's lots of hedgehog made in the posterior compartment and then less of it here. Like that, okay? Because it's made here, it's spreading that way. So that's that concentration gradient. And then if we did this for Wnt, we'd see lots of Wnt right here at the compartment boundary and then less of it, okay? And it just goes over a short range. On the other hand, and I guess I used DPP for this, DPP is made here and it spreads in both directions and it spreads further. It spreads all the way across the segment, even when the segment starts to get really big. When the segment starts to get really big, these short-range signaling factors are just limited to the cells right along that compartment boundary, but DMP continues to spread across the whole boundary in both directions. And DPP helps pattern that overall segment. For example, there's going to be a wing maybe on this segment, so that could be an insect wing. And the insect wing has some sensory bristles along the anterior edge of the wing. And those help detect the air. The little bristles are like little feelers that help detect the air as the wing is moving. And DPP represses the bristle formation. So we don't see any bristles in the middle of the wing, they're just at the anterior edge. And so that's an example of DPP as a long-range signaling factor. And I put a couple more numbers down here. I think that's a really good list for your assignment. So this is what your assignment should look like, and not as messy, but make it big. Even bigger than this is great, and you just have to tape some pieces of paper together or make it in two, like that. But what you should do in your assignment is you should include the drawing and label, and then this list. And you can make the list, off to the side or just on your computer like that. But this list, and you should know and explain, what these components are. I added a couple more numbers here because there are more things that we could add, and maybe we will, but not for the assignment. Maybe we'll add them as we talk, or there are questions in class. Something else will come up. And so, what does it mean to be a posterior cell? It means to express a certain set of genes. What genes? Well, we saw in the muscle. Muscle expresses genes like actin and myosin that make the muscle fiber. And we saw in the mesoderm that mesoderm expresses transcription factors like brachyury that help to activate genes that are on the pathway to making muscles and bones. And we saw in induced pluripotent stem cells, we misexpress some transcription factors, and they activate cells that are only expressed in stem cells and cause cells to become stem cells. So here, to be a posterior cell, we need to express this. The frizzled receptor to be hearing, be a posterior cell, and activate posterior compartment genes, right? That's beta-catenin's role. Now, we've seen beta-catenin over and over again because it's another one of these context-dependent transcription factors, one of these developmental regulators that is like a wrench, and it has different target genes in different cellular contexts. So here, beta-catenin is helping cells become posterior cells. And so it's helping activate frizzled, and posterior cells need to express hedgehog. And that's fascinating because the role of hedgehog is to tell anterior cells to be anterior. That's the only role of it. It's not really a role for posterior cells, except posterior cells want to create a kind of otherness, like we are we, and we want somebody else to be them on the other team. Okay? Then the set of transcription factors, engrailed is one, that's going to help activate a variety of posterior specific cells. And then the new story, these cell adhesion molecules, which are fascinating and used, hugely throughout our body and throughout all animal development to help create difference, right? We want one of the things in creating a multicellular animal is to create patterning, cells becoming different. And so cell adhesion molecules help cells become different by segregating them, and keeping those populations separate. And when they are separate, they can develop more and more different roles, like some anterior wing edge bristles. And then our last one is that this boundary and then allows the developing segment to have a landmark, like a survey line that says this is the compartment boundary, and we'll have different things happening in the anterior posterior compartments, like anterior bristles. And there are long-range signaling factors like DPP that are expressed in the posterior and spread long distances to help with the overall patterning. Okay, great. And so that's our assignment. Woohoo! For drawing. Okay, and thanks.